Okay, so welcome everybody. Really happy Friday. So everybody, I'm sure, is happy that it's Friday, and today is day 15. So just a reminder, we have our lab reports due on Monday, so you're definitely going to want to put some time in to make sure you get those done for Monday, because I will be charging you a late 10% per day. Okay. Again, it's important to establish those deadlines. So what we're going to do today is first going to do a little take a look at some of that summary that I gave you the other day, and just take up some key points. So if we go to our content section here. And I'm just going to go back to Apple here, and just we're going to put this on top so you can see me the whole time. So let's take a look at our first summary. Okay, so some terminology. I'm not going to go through with what each term means. You can look that stuff up. But make sure the key thing is that you can use it in a sentence. And what I mean by that is when you're talking about, say, monosaccharides and disaccharides, I know that you can just look up the definition of a monosaccharide is the following, and a disaccharide is the following, but being able to use it in a sentence such as monosaccharides combine in order to form disaccharides kind of lets you know that you actually understand the term, as opposed to just this is the definition of a monosaccharide, this is the definition of a disaccharide. Okay, so let's go down here. I'm just going to move myself up. So, what are all living things composed of? I'm assuming you said a cell, and then what are cells made up of? They're made up of our four macromolecules. Okay, what elements are used to create the cell? We should know the CHNOPs. Okay, so CHNOPS, so carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Okay, what do functional groups do to a molecule? Well, functional groups simply provide function. Now, that's not really the best answer, but they really provide different ways that the molecule can now interact. It can provide polarity. It can provide non-polarity. It basically takes a carbon backbone and then adds function to it by adding these functional groups. What are the four building blocks of life? There are four macromolecules, okay? Carbs, lipids, proteins, and nucleic acids. Why do we need to eat? Now, this is a very good question because everyone always answers it one way. We need to eat because we need to have energy. And you're right, okay? But again, like I mentioned before, you also need to build new cells, okay? As you grow, as you repair yourself, as you perform mitosis and meiosis, those things we talked about in grade 11, you need to have the building blocks in order to do that. So you eat so that you have a carbohydrate or a fat so that you can build a new cell. Okay? So then we get into carbohydrates, our first macromolecule. What are the three monosaccharides that make up most carbs? So you should know glucose, fructose, and galactose. Okay, those are our three monosaccharides. Okay, and then when we look at maltose, so maltose is just going to be two of those combined together. Okay, lactose is also going to be that. So lactose is glucose, and it's going to have another one. So lactose is glucose and galactose combined together. Sucrose is glucose and fructose. Okay, and then maltose is going to be two glucoses. So you should know that. That, the three monosaccharides and then what is composed of each of these three. So what's the basic structure of a carb? You're going to need to have 6 carbons, 12 hydrogens, and 6 oxygens. All carbohydrates are that C6H12O6. Okay, and the elements are going to be carbon, hydrogen, and oxygen. Those are the only 3 elements. There's no phosphorus, there's no sulfur, there's no nitrogen. If you're seeing those, they're not a carbohydrate. All right, what are the functions of carbs in living things? There's 3 main functions. We've got immediate energy, like our blood glucose levels. We've got medium to short-term energy, which would be like glycogen, okay, or starch. And then our third one, what everybody forgets, is structure. Okay, we use carbohydrates for structure, like a plant does. Creates its basically body out of cellulose, which is just a whole bunch of sugar molecules all connected together. Okay? So the difference between cellulose, starch, and glycogen. So cellulose is going to be mostly just glucose, fructose, maltose. It's going to be a variety of monosaccharides all linked together. Starch and glycogen are very, very similar. Okay, they're going to be purely glucose molecules. And starch is going to be the way that plants store their medium energy, and glycogen is how animals store their medium energy. But other than that, they're pretty much exactly the same, just 2 different organisms storing their medium energy 2 different ways. Okay, and then to test for simple carbs, that was the lab that you did. So it's the Benedict test. And if the Benedict's basically a blue solution, so if the solution that you're suspecting has sugar in it remains blue, it doesn't. And if it turns to like an orangey red color, then you know it has simple carbohydrates. Okay, not complex carbohydrates, simple carbohydrates. For complex carbohydrates, like a polysaccharide, like cellulose or starch, or glycogen. In order to test for that, we use iodine. So iodine has a kind of a natural yellowish color. If it reacts with a complex carb, then it turns a black color. So those are our two tests for those. Okay. So protein, the role of proteins in the body. Okay, it has a ton of different roles in the body. Such things as our immune system, for structure, for enzymes, for protein channels. There's a variety of different functions for protein. What is a polypeptide and what are our subunits? A polypeptide is just a series of amino acids all connected together. So, you're going to have basically, let me just draw this out quickly. So I'll put this up to the screen here. So this is a polypeptide. These would be individual amino acids all connected together. Okay, and this long chain is going to create a protein. So what are the subunits? They're amino acids. The basic structure of amino acids, you should know that. Okay, it's in your content. And what elements are in there? You're going to have carbon, hydrogen, oxygen. You're also going to have sulfur in there. Okay, so this is the first time we see sulfur and phosphorus as well. Okay. An important protein is enzymes, so you should know what enzymes are. They are basically catalysts of reactions. Very important, but we'll talk about those a little bit later on in the course. You should know that shape is a key factor for protein function. So proteins work based on their shape. If you change their shape, they become inactive. And that's why when we heat up things to destroy microorganisms, we're heating them up to change the shape of their proteins, which deactivates them. And that's why we boil water to kill all the bacteria, because when we boil it, we denature the bacteria's proteins and then destroy them. Okay. How do we test for protein? So protein is, and I may be pronouncing this wrong, is the Biuret test. Okay, and it's also another blue liquid, and it's going to change color if you get a positive test for protein. Lipids, our last one. So the purpose of lipids in the body, okay, they're kind of our long-term storage. They're also insulation. They're also cushioning. So they also have a variety of functions. They should be able to draw the basic structure of a triglyceride. So the fact that we have a glycerol and then 3 fatty acids attached to it. Okay, a phospholipid, same thing. We got a glycerol, but instead of 3 fatty acids, we're going to remove 1 fatty acid and put a phosphate group on there, and that's a phospholipid. And then hormones. Okay, that you should know that fats are used for hormones like testosterone and estrogen, but I'm not going to ask you to know the structure of those. Okay. Why would they be used as waterproofing in the body? So I got a couple questions about this. So fats don't interact with water at all. Okay, so that's how we basically waterproof things. So if we wanted to make a waterproof membrane, we could use plastic because plastic doesn't interact with water. Well, our bodies can't make plastic. Okay, but we can make something pretty similar to plastic, which is fats. Okay, and we use these to basically waterproof any area that we need. When we want to create a cell, we want to keep all of those components inside the cell. We want to make sure the mitochondria is left in there, the nucleus in there, it doesn't basically escape out. Well, in order to hold all of that stuff together, okay, we use basically a layer of fat around the outside, which we call our plasma membrane, and those are made up of phospholipids. We're going to talk a lot more about that in our second chapter. Okay. How do we test for lipids? We use Sudan-4. Okay, and when you went through your lab, you'll know the positive test for that. It's basically little droplets of color inside the solution. Okay, nucleic acids, the basic structure of a nucleotide. So you have your base, your sugar, and your phosphate. Okay, those 3 things all connected together. What is DNA used for? Okay, it's used to basically coding for the construction of a body as well as running the body. So we use our DNA to tell the cells on basically how to construct things as well as after they're constructed, how to run them properly. The differences between RNA and DNA, okay, I kind of went over a little slideshow on that. You should know the differences between the two. There's 3 major ones. And how would you know if nucleic acids were in your food? So this is kind of a tricky question, but I don't think we ever went over this. But there's not a test for nucleic acids. The FDA doesn't really test for nucleic acids. But you do know if you have nucleic acids in your food. So I want you to think about this for a second. Okay? Would a glass of Kool-Aid have nucleic acids in there? Would a glass of milk have nucleic acids in there? Okay. Now, one of them is and one of them doesn't. Now, if you guessed milk has nucleic acids, you're right. And the reason why milk will have it is there'll be cells inside the milk. And anytime you have a cell, you must have nucleic acids. But when we talk about, say, something like a glass of Kool-Aid, there's sugar in there. There's macromolecule sugar, but there's not cells in there. There's not entire cells. So if there's not an entire cell, there's not going to be nucleic acids or DNA. Okay? So anytime you eat a cell, like you eat a piece of chicken, you eat a vegetable or a fruit, as long as you're eating a living thing, okay, then you must be getting nucleic acids. Okay? So that was our Activity 1 summary. Now, if we go back to content, we're going to start our new chapter today, and our next new chapter now is going to be basically how to run a cell. So we originally talked about building a cell with our macromolecules. Now we're going to be talking about running our cell. So we're going to be going on to Activity 2, or Chapter 2 in the textbook. So you can click on this if you like, and it gives you kind of an overview, and those are my expectations. The one that we're going to be covering today is Content 1, which is basically just the cell. And I know this can be kind of dry on basically how a cell runs, so I'd like you to look through this content, okay, and go through this content. There's a bunch of information here. But this is the big thing I'd like you to do today, is the assignment. Now, I'm going to make this assignment not really have a set deadline until we're done this chapter, or done Activity 2. So it'll probably be due, I'll put it in there, probably about a week or something like that. Okay, I really want you to focus on your lab report. This assignment's very key, though. Okay, when we have to memorize a lot of information as biologists, and we do a lot because biology is a lot of memory work, it's difficult. It's difficult to remember, what does the Golgi apparatus do? What does the mitochondria do? What does the plasma membrane? Okay, it is a very difficult task to memorize the meaning behind all of these. So we have these little tricks that we do. Okay, and if you go on to med school, or if you go into things where you have to memorize a ton of information, it's difficult to just say, okay, I'm going to try and read it and memorize it. A better way is to try and use kind of little memory tricks that we do. And one of them is basically creating an analogy. And for the cell, a very key thing is to make an analogy. And what I like to do is make an analogy of a city. And when you watched our video, so if we go back to our home screen here, and I love this guy, okay, this guy right here, Crash Course, and he talked about how a animal cell can be considered a city. Okay, and he did 2 videos in here. He's really entertaining. Okay, that's why I posted his videos. He's way more entertaining than myself. And you know, again, he has videos for all of our content, so you can look those up. I'll post a lot of them. But if we go back here, what I'd like you to do today, or not today, but I mean get started on in the next couple days, is this assignment. And it's assignment 1. And basically what you have to do is create an analogy of how a cell is like a city. So you can format this any way that you like, but how I would like to format, or if I was me doing it, this is how I would format it. But again, you can present this in any way that you like. Okay? It's having basically, okay, if we have a mitochondria in the cell part, so I'd probably do a table, cell part, mitochondria, analogy of human part, power plant, and then an explanation as to why the two are similar. So mitochondria is very similar to a power plant in a city, because in a city, the power plant provides, you know, energy for the city, based taking raw materials and turning it into energy. That's what a mitochondria does. It takes raw materials, glucose, and turns it into energy, ATP, for the cell. Okay. Now, the only parts that you're going to need to know for the cell, because there's a lot more parts, are the ones that I have listed in here. Okay. So things like, starting here, liposomes, peroxisomes, vacuoles, mitochondria. So you don't need to do every part of the cell. It's just these parts right here. Okay, where I'd like you to create an analogy. And then once you remember this city, it's going to help you remember, you know, what different parts of the cell do. Oh, what does the Golgi apparatus do? Oh, yeah, it's like the post office. Okay, and it'll help you remember these things. So we're not just doing this to remember the content. I'm hoping that you develop a skill that you can use in the future, that when you have to memorize hard things, creating analogies. And then I'd like you to answer these questions. Now, again, the due date on here, I will post, and it's probably going to be about a week. So probably next Friday is when I'd like to get this thing in. I'm going to have other things due in between that, but this isn't essential for us to move on. Okay? It's helpful, but it's not essential for us to do further content. So that's what I'd like to work on today. Hope you guys have an excellent weekend. And again, I'm going to have this little pause at the very end here so that, you know, I don't get cut off.